Inside human renewal, from cellular repair to sleep, emotion, and neuroplasticity. Welcome to Dream Lab Knowledge, where we explore the fascinating science of life, consciousness, and everything that makes us human. Tonight, we're embarking on an extraordinary journey into the hidden world of human renewal, discovering how your body rebuilds itself while you rest, how your brain rewires itself as you learn, and how the simple act of sleeping becomes the most profound form of self-care. Close your eyes for a moment and feel the rhythm of your breath. In this very instant, within the quiet sanctuary of your body, millions of invisible workers are engaged in the most extraordinary maintenance project ever conceived. Cells are repairing themselves, damaged proteins are being recycled, neural pathways are rewiring themselves, and an ancient biological clock is coordinating it all with precision that would humble the finest Swiss watchmaker. You are, in the truest sense, rebuilding yourself moment by moment, breath by breath. This is the story of human renewal, a journey into the hidden architecture of restoration that operates beneath your awareness, transforming you while you sleep, while you think, while you feel. It is a story written in the language of molecules and neurons, hormones and electrical signals, yet it speaks to something profoundly human: the remarkable capacity of your body to heal, adapt, and regenerate. The silent repair crew. Imagine your body as a vast city that never sleeps. Within this metropolis, each cell is both a building and a resident, simultaneously structure and inhabitant. Like any city, wear and tear is inevitable. Sunlight damages DNA like weather eroding stone. Free radicals from metabolism attack cellular membranes like rust corroding metal. Proteins misfold and aggregate like debris accumulating in alleyways. Yet, unlike any human city, this one possesses an extraordinary feature. It can repair itself from within, using mechanisms so sophisticated that scientists are only beginning to understand their full complexity. At the heart of cellular repair lies DNA, the blueprint that contains the instructions for building and maintaining every structure in your body. DNA experiences damage constantly. Ultraviolet radiation from sunlight can create abnormal bonds between adjacent thymine bases, forming what scientists call thymine dimers. Reactive oxygen species, the byproducts of normal metabolism, can modify DNA bases or create breaks in the double helix. Even the simple act of DNA replication, necessary for cells to divide, introduces errors at a rate of about one mistake per billion base pairs copied. If left unrepaired, these errors would accumulate catastrophically. A human cell experiences tens of thousands of DNA lesions every single day. Over a lifetime, without repair, mutations would pile up like snow in an avalanche, overwhelming cellular function and leading to cancer, aging, and death. Yet, you survive, you thrive, because your cells possess an arsenal of repair mechanisms that patrol the genome ceaselessly, finding and fixing damage with remarkable efficiency. The most elegant of these repair systems is called base excision repair. Imagine specialized enzymes as molecular detectives, moving along the DNA double helix, feeling each base pair with chemical fingers, searching for abnormalities. When they encounter a damaged or incorrect base, these enzymes, called DNA glycosylases, clip it out of the DNA strand, leaving a gap. Other enzymes recognize this gap, a signal that repair is needed. DNA polymerase fills in the missing piece, using the complementary strand as a template, and DNA ligase seals the final connection, restoring the integrity of the genetic code. The entire process takes mere seconds, yet it happens thousands of times in each cell every day. For more severe damage, such as double-strand breaks, where both strands of the DNA helix are severed, cells employ two major strategies. Homologous recombination uses the undamaged sister chromosome as a template to accurately rebuild the broken region, like copying text from a backup document. Non-homologous end joining, faster but less precise, simply glues the broken ends back together. The choice between these pathways depends on the cell cycle stage and the nature of the damage. A decision made by molecular machinery that evaluates the situation and responds accordingly. But DNA is not the only target of cellular damage. Proteins, the workhorses of the cell, are equally vulnerable. Heat can cause them to unfold. Oxidative stress can damage their chemical structure. Simply aging in the cellular environment can cause proteins to lose their proper three-dimensional shape, the conformation that determines their function. A misfolded protein is like a tool that has lost its edge, unable to perform its intended task, and worse, misfolded proteins can aggregate together, forming toxic clumps that interfere with cellular processes. Enter the molecular chaperones, proteins whose entire purpose is to help other proteins fold correctly. Heat shock proteins, named for their increased production during cellular stress, grab onto partially unfolded proteins and give them another chance to fold properly. If refolding fails, these chaperones can direct the damaged protein to the cellular recycling system. Some chaperones, like the remarkable protein called chaperonin, form a barrel-shaped chamber where misfolded proteins can be enclosed and given a protected space to refold, isolated from the crowded cellular environment that might cause further aggregation. When proteins are beyond saving, the cell marks them for destruction by attaching small protein tags called ubiquitin. These tags are like shipping labels that direct damaged proteins to the proteasome, a cylindrical protein complex that functions as a cellular shredder. The proteasome unfolds and feeds proteins through its central channel, where they are chopped into small peptides that can be recycled into new proteins. This ubiquitin proteasome system processes thousands of proteins every minute in each cell, maintaining the delicate balance between protein synthesis and degradation. The great cellular cleanup beyond the proteasome lies an even more powerful recycling system, one that can consume not just individual proteins, but entire organelles, a process called autophagy, literally meaning self-eating. Autophagy is the cell's way of clearing out the old and damaged, making room for the new, and in doing so, it plays a crucial role in health, longevity, and disease prevention. Picture autophagy as a cellular waste management system of exquisite sophistication. When a cell experiences stress, nutrient deprivation, or accumulation of damaged components, it initiates this cleanup program. A cup-shaped membrane called a phagophore begins to form, extending its edges like closing hands around the cellular debris that needs to be removed. This might be a cluster of misfolded proteins, a damaged mitochondrion, or even invading bacteria. The phagophore gradually elongates and closes, forming a double membrane vesicle called an autophagosome, which now contains the condemned cellular components. The autophagosome is not static. It travels through the cell, migrating toward the perinuclear region, where lysosomes cluster. Lysosomes are organelles filled with powerful digestive enzymes capable of breaking down virtually any biological molecule. When an autophagosome fuses with a lysosome, it becomes an autolysosome, and the contents within are degraded into their constituent parts: amino acids from proteins, fatty acids from lipids, sugars from carbohydrates. These building blocks are then released back into the cytoplasm, where they can be reused to build new cellular structures or burned for energy. Autophagy is not a single process, but a family of related mechanisms, each specialized for particular targets. Mitophagy specifically removes damaged mitochondria, those power plants of the cell where energy is generated, but where reactive oxygen species are also produced as toxic byproducts. When a mitochondrion becomes dysfunctional, its membrane potential drops, triggering a cascade of molecular events. Proteins like PINK1 accumulate on the mitochondrial surface, recruiting another protein called parkin, which decorates the mitochondrion with ubiquitin tags. These tags serve as an "eat me" signal, attracting autophagy receptors that link the damaged mitochondrion to the autophagy machinery, ensuring its removal before it can poison the cell. The importance of mitophagy becomes starkly clear in neurodegenerative diseases. In Parkinson's disease, mutations in PINK1 or parkin prevent proper mitophagy, allowing damaged mitochondria to accumulate in neurons. These dysfunctional mitochondria produce excessive reactive oxygen species while failing to generate adequate energy, leading to neuronal death and the characteristic motor symptoms of the disease. The selective death of dopamine-producing neurons in the substantia nigra, the hallmark of Parkinson's, is intimately connected to failures in this quality control system. Autophagy declines with age, a fact that carries profound implications for human health and longevity. Studies in various organisms, from yeast to mice to humans, show that autophagy becomes less efficient as we grow older. Autophagy-related genes are expressed at lower levels. The formation of autophagosomes slows. The fusion between autophagosomes and lysosomes becomes less efficient. The result is an accumulation of cellular damage, protein aggregates, dysfunctional organelles, oxidative stress, all the molecular garbage that cannot be properly cleared. This age-related decline in autophagy contributes to many hallmarks of aging. In the brain, reduced autophagy allows misfolded proteins to accumulate, contributing to Alzheimer's disease, where beta amyloid plaques and tau tangles poison neural circuits. In muscles, declining autophagy contributes to sarcopenia, the loss of muscle mass and strength that affects the elderly. Throughout the body, autophagy's weakening allows senescent cells to persist. These zombie-like cells that have stopped dividing but refuse to die, secreting inflammatory molecules that damage their neighbors. Yet the decline of autophagy is not inevitable or irreversible. Research in model organisms has shown that stimulating autophagy can extend lifespan and improve health during aging. Caloric restriction, the most robust intervention known to extend lifespan in diverse species, works in part by activating autophagy. When nutrients are scarce, cells upregulate autophagy to recycle their own components for energy and building materials, a process that simultaneously performs housekeeping duties, clearing out accumulated damage. Intermittent fasting produces similar effects, giving cells regular periods when autophagy is heightened. Exercise also stimulates autophagy, particularly in muscle tissue, where the physical stress of contraction triggers cellular cleanup. Even certain compounds can activate autophagy pathways. Rapamycin, a drug that inhibits the mTOR signaling pathway, a key negative regulator of autophagy, has been shown to extend lifespan in mice and improve multiple age-related pathologies. Spermidine, a natural compound found in foods like aged cheese, mushrooms, and wheat germ, can also induce autophagy and has been associated with increased longevity in multiple organisms. The membrane repair emergency. While DNA repair and autophagy work on timescales of minutes to hours, some cellular repairs must happen in seconds, or the cell will die. Consider the plasma membrane, the thin lipid barrier that separates a cell's interior from the outside world. This membrane is constantly under assault. Mechanical forces can tear it. Pathogens can puncture it. Even normal cellular activities can damage it. When the membrane ruptures, the cell faces catastrophe. Cytoplasm spills out, essential ions rush in, and the delicate chemical gradients that power cellular processes collapse. Yet cells can survive such breaches through rapid membrane repair mechanisms that seal holes within seconds. When the plasma membrane is damaged, calcium ions from outside the cell rush through the breach. This sudden spike in calcium concentration acts as an alarm signal, triggering repair machinery. Within seconds, nearby vesicles, small, membrane-bound bubbles, waiting in the cytoplasm, are recruited to the damaged site. These vesicles fuse with each other and with the torn edges of the membrane, like biological spackle filling a crack in a wall. The speed is astonishing. Studies using high-speed microscopy have shown that cells can reseal puncture wounds in as little as 3 to 30 seconds. This rapid response is essential because every second of delay allows more cytoplasm to leak out. and more potentially toxic substances to flood in. The membrane repair machinery must be constantly ready, poised to respond to damage that could occur at any moment. Different cell types face different membrane challenges and have evolved specialized repair mechanisms. Heart muscle cells, which contract billions of times over a lifetime, routinely experience mechanical stress that damages their membranes. They possess enhanced membrane repair capabilities, including higher levels of repair proteins and more robust vesicle recruitment systems. When these systems fail, as can happen in certain muscular dystrophies, even normal contractions can cause progressive damage, leading to muscle degeneration. Neurons face unique membrane challenges because of their extreme morphology. A single neuron can extend processes, axons, and dendrites that are thousands of times longer than the cell body is wide. Maintaining membrane integrity across such vast distances requires specialized mechanisms. Damage to an axon, far from the cell body, must be detected and repaired locally without waiting for signals to travel back to the nucleus and return. Neurons accomplish this through local stockpiles of membrane repair components distributed along the axon, ready to respond instantly to damage. Tissue regeneration and the limits of renewal. While individual cells possess remarkable repair capabilities, the renewal of tissues and organs operates on a different scale, involving coordination between millions of cells. Some tissues renew themselves constantly throughout life. Others have limited regenerative capacity. Still others, once damaged, cannot regenerate at all, at least not in adult mammals. The intestinal lining represents one extreme of regenerative capacity. The epithelium that lines your intestines, exposed to harsh digestive enzymes and potential pathogens, is completely replaced every 3 to 5 days. At the base of intestinal crypts, small pits in the lining, stem cells divide continuously. Some daughter cells remain as stem cells, maintaining the population. Others begin to differentiate, migrating up the sides of the intestinal villi, those finger-like projections that increase surface area. As they travel, they mature into specialized cells, absorptive enterocytes that take up nutrients, mucus-secreting goblet cells, hormone-producing enteroendocrine cells. By the time they reach the tips of the villi, they are fully differentiated and functional. Soon after, they undergo programmed cell death and are shed into the intestinal lumen, replaced by the constant stream of new cells from below. This continuous renewal is essential because of the harsh environment these cells inhabit. Digestive enzymes that break down food also attack cellular proteins. Bacteria in the gut lumen could invade if the barrier were compromised. The rapid turnover ensures that damaged cells are quickly replaced before they can fail in their protective duties. Your skin employs a similar strategy, though with a longer replacement cycle. The outermost layer of your skin, the epidermis, is constantly renewed by stem cells in the basal layer. These cells divide, and their daughters migrate outward, gradually transforming as they go. They accumulate the tough protein, keratin, flatten out, and eventually die, becoming the protective dead cell layers that form your body's outer shield. These dead cells are continuously shed, about 30 to 40,000 every minute, replaced by new cells from below. The entire epidermis renews itself approximately every 4 to 6 weeks. The liver, too, possesses extraordinary regenerative capacity. If up to 70% of the liver is removed, the remaining tissue can regenerate the lost mass within weeks. This regeneration is not true regrowth in the sense of rebuilding the exact same structure that was lost. Rather, the remaining liver cells, normally quiescent, begin to divide rapidly, increasing their numbers until the liver reaches its original size. The hepatocytes, liver cells, then return to their resting state. This regenerative capacity makes liver transplantation possible, both from deceased donors and from living donors who can give a portion of their liver knowing it will regenerate. Yet, not all tissues share this regenerative prowess. Heart muscle, once damaged, has minimal capacity to regenerate. When a heart attack kills cardiac muscle cells, the dead tissue is replaced by scar tissue, non-contractile collagen that fills the gap but cannot contribute to the heart's pumping function. The surviving heart muscle must work harder to compensate, often leading to progressive heart failure. This limitation has puzzled scientists for decades. Why should the heart, so vital to survival, lack the regenerative capacity of tissues like skin or intestine? Part of the answer lies in the nature of cardiac muscle cells. Unlike intestinal or skin cells, which divide frequently, adult cardiac muscle cells have withdrawn from the cell cycle. They no longer divide. This makes sense in one way. A heart cell that stopped contracting to divide would create a gap in the heart's pumping action. But it creates a dilemma when damage occurs. Recent research has revealed that the heart is not completely unable to regenerate, just very limited. A small number of cardiac muscle cells do divide in adult humans, perhaps 1% per year at age 20, declining to less than 0.5% by age 75. This minimal turnover is insufficient to repair major damage, but suggests that the heart retains at least some regenerative machinery. More intriguingly, newborn mammals, including mice and even humans, can regenerate heart tissue for a brief window after birth. If a portion of a newborn mouse's heart is damaged within the first week of life, the heart can completely regenerate the lost tissue. This capacity is lost within days as the cardiac muscle cells exit the cell cycle and commit fully to their contractile function. Understanding why this regenerative capacity is lost and whether it could be reactivated is a major focus of regenerative medicine research. Some researchers are investigating the molecular signals that keep adult cardiac muscle cells from dividing, hoping to transiently reactivate cell division to repair damaged hearts. Others are exploring whether stem cells, either from the patient or created in the laboratory, could be coaxed into becoming new cardiac muscle cells that could integrate into damaged heart tissue. Neurons present another regenerative challenge. Most neurons in the adult human brain do not divide. Once formed during development, they must last a lifetime, often more than a century in individuals who live to advanced ages. When neurons die, whether from stroke, trauma, or neurodegeneration, they are generally not replaced. This has profound implications for brain injury and disease. There are exceptions. The hippocampus, crucial for forming new memories, continues to generate new neurons throughout life, a process called adult neurogenesis. Several thousand new neurons are born every day in the human hippocampus, integrating into existing circuits. This neurogenesis is essential for certain types of learning and memory formation, and it can be modulated by experience. Exercise, enriched environments, and learning new skills all increase hippocampal neurogenesis, while chronic stress, depression, and aging reduce it. In the peripheral nervous system, neurons can regenerate damaged axons, the long processes that carry signals to distant targets. If a peripheral nerve is cut, the portion of the axon separated from the cell body will degenerate, but the portion still connected to the cell body can regrow, extending a new axon toward its original target. This regrowth is slow, typically 1 to 3 millimeters per day, and requires a permissive environment. Supporting cells, called Schwann cells, in the peripheral nervous system create a pathway for the regrowing axon to follow, like a tunnel guiding the regenerating nerve back to its target. In contrast, the central nervous system, the brain and spinal cord, actively inhibits axon regeneration. When axons in the brain or spinal cord are damaged, they typically do not regrow. Multiple factors contribute to this failure. Inhibitory molecules in the central nervous system, myelin, the insulating sheath around axons, actively prevent regeneration. Scar tissue that forms after injury creates a physical and chemical barrier. The neurons themselves may have reduced intrinsic capacity for growth compared to peripheral neurons. This difference between peripheral and central nervous systems explains why crushing a peripheral nerve can eventually heal, while spinal cord injuries result in permanent paralysis. It also drives intense research into strategies to overcome central nervous system inhibition of regeneration, from neutralizing inhibitory molecules to providing growth-promoting factors to engineering scaffolds that guide axon regrowth across injury sites. The architecture of sleep, while cellular and tissue repair mechanisms operate continuously, many repair processes are enhanced during sleep, that mysterious state that occupies roughly 1/3 of human life. For most of human history, sleep was considered a passive state, a shutting down of consciousness that served primarily to conserve energy and keep us safe during the dangerous night hours. Nothing could be further from the truth. Sleep is an intensely active state, a time when the brain and body engage in essential maintenance activities that cannot be adequately performed during waking. Sleep is not uniform, but consists of distinct stages that cycle through the night. When you first drift off, you enter stage 1 of non-rapid eye movement sleep, a brief transition between wakefulness and sleep, where you can be easily awakened. From there, you descend into stage 2, characterized by sleep spindles and K complexes, brief bursts of brain activity whose functions are still being understood but appear related to memory consolidation and sensory processing. Then comes stage 3, deep sleep or slow wave sleep, named for the large, slow delta waves that dominate the electroencephalogram. This is the most restorative stage of sleep, the time when your body's repair systems operate at peak efficiency. Blood flow to the brain decreases, while blood flow to muscles increases, delivering oxygen and nutrients needed for tissue repair. Growth hormone is released in its greatest quantities, stimulating tissue growth and repair throughout the body. The immune system becomes particularly active, with production of cytokines and mobilization of immune cells. After progressing through these stages of non-REM sleep, you enter REM sleep, named for the rapid eye movements that occur during this stage. In REM sleep, your brain activity increases dramatically, approaching waking levels, while your body becomes paralyzed to prevent you from acting out dreams. This stage is crucial for emotional regulation and memory consolidation, particularly for procedural memories, the kind of memories involved in learning new skills. These stages cycle through the night in roughly 90-minute periods. In the first half of the night, deep non-REM sleep predominates, giving your body ample time for physical restoration. In the second half of the night, REM periods become longer and more frequent, providing extended time for mental and emotional processing. This architecture is not arbitrary, but reflects the different functions served by each stage. The restorative functions of sleep operate at multiple scales. At the cellular level, sleep enhances DNA repair mechanisms. Studies in sleep-deprived rats show dramatically increased DNA damage in multiple organs, particularly the brain, liver, and lung. When these animals are allowed to sleep, DNA repair mechanisms spring into action, reducing damage back to normal levels within 2 days. The mechanisms behind this enhanced repair during sleep are still being elucidated, but they likely involve increased expression of DNA repair enzymes and reduced oxidative stress. Protein synthesis also increases during sleep, particularly in the brain. Throughout the day, neural activity generates proteins that need to be replaced, or new synaptic connections that need to be strengthened. Sleep provides the time for this molecular construction work. Studies have shown that preventing sleep after learning a new task impairs the protein synthesis needed to consolidate that learning into long-term memory. Autophagy, that cellular cleanup system discussed earlier, operates on a circadian rhythm with activity peaking during sleep. The brain has limited capacity for autophagy during wakefulness because neurons are busy processing information, firing action potentials, releasing neurotransmitters. During sleep, particularly deep sleep, neural activity becomes more synchronized and less demanding, creating windows where autophagy can proceed more efficiently. This nighttime cellular housekeeping removes damaged proteins and organelles that accumulated during the day's mental exertions. The glymphatic system, one of the most remarkable discoveries about sleep in recent years, involves a waste clearance system in the brain called the glymphatic system, named for its dependence on glial cells and its functional similarity to the lymphatic system. that drains waste from other body tissues. Unlike other organs, the brain lacks a conventional lymphatic system. For decades, how the brain removed metabolic waste products was unclear. The answer turns out to be intimately connected with sleep. During waking, the brain is densely packed with minimal space between neurons and supporting cells. This tight packing facilitates rapid communication between neurons, but leaves little room for fluid movement. During sleep, particularly deep sleep, something remarkable happens. The brain cells shrink slightly, increasing the volume of extracellular space by as much as 60%. This expansion creates channels through which cerebrospinal fluid can flow. Cerebrospinal fluid, the clear liquid that bathes the brain and spinal cord, enters the brain along channels that surround blood vessels, flowing into the extracellular space. As it percolates through the brain tissue, it collects waste products, beta amyloid, tau, metabolites from neural activity, all the molecular debris generated by the brain's incessant work. The fluid then drains out along different channels, carrying the waste products with it to be filtered through the regular lymphatic system and eventually excreted. This system is dramatically more active during sleep than during waking by as much as 10 to 20 times. The increase in extracellular space during sleep is crucial for allowing adequate fluid flow. When sleep is disrupted or insufficient, the glymphatic system cannot adequately clear waste products, and they begin to accumulate. The implications for neurodegenerative disease are profound. Beta amyloid, the protein that forms plaques in Alzheimer's disease, is produced continuously by neurons as a byproduct of normal activity. In healthy individuals, it is cleared by the glymphatic system before it can accumulate to toxic levels. When sleep is chronically disrupted, beta amyloid clearance decreases and the protein begins to build up. This accumulation can, over years or decades, contribute to the development of Alzheimer's disease. The relationship appears to be bidirectional and vicious. Sleep disruption leads to beta amyloid accumulation, and beta amyloid accumulation disrupts sleep, creating a downward spiral. Brain regions important for sleep regulation are among the first affected by Alzheimer's pathology, explaining why sleep disturbances often appear years before memory problems become apparent. This has led researchers to investigate whether improving sleep could be a strategy for preventing or slowing Alzheimer's disease. Studies in animal models support this possibility. When researchers experimentally increased glymphatic clearance, they found reduced beta amyloid accumulation and improved cognitive function. In humans, observational studies have found that people who report chronic sleep problems have higher rates of developing dementia years later, even after controlling for other risk factors. While correlation does not prove causation, the mechanistic understanding of the glymphatic system provides a plausible explanation for this association. The glymphatic system clears other waste products as well. Tau, another protein that aggregates in Alzheimer's disease and other neurodegenerative conditions, is removed via glymphatic clearance. Lactate and other metabolites from neuronal activity are flushed out. Even neurotransmitters may be cleared through this system, helping to reset neurochemical balances for the next day. This nightly brainwashing is yet another reason why sleep is not optional for brain health. You cannot simply push through on coffee and willpower without consequences. Each night of inadequate sleep means another day's worth of metabolic waste not properly cleared. Another increment of damage that accumulates invisibly until years later it manifests as cognitive decline or neurodegenerative disease. Hormonal rhythms of restoration. While the glymphatic system operates on the time scale of sleep stages, other restorative processes in the body are governed by circadian rhythms, biological oscillations with periods of approximately 24 hours that synchronize physiological processes with the day-night cycle. At the center of this system is a tiny cluster of neurons in the hypothalamus called the suprachiasmatic nucleus, your brain's master clock. The suprachiasmatic nucleus receives direct input from specialized light-sensitive cells in the retina, allowing it to synchronize with the external light-dark cycle. When daylight hits these retinal cells, signals travel to the suprachiasmatic nucleus, which responds by sending signals throughout the brain and body that say, "It is day." As light fades in the evening, the suprachiasmatic nucleus responds by triggering the release of melatonin from the pineal gland, a small structure deep in the brain. Melatonin is often called the sleep hormone, but its role is more accurately described as a signal of darkness. Melatonin levels in the blood begin to rise in the evening, peak in the middle of the night, and fall as morning approaches. This melatonin rhythm helps to promote sleep, but it also coordinates circadian rhythms throughout the body, synchronizing peripheral clocks in organs like the liver, heart, and kidneys. These peripheral clocks are molecular oscillators. present in nearly every cell of your body. They consist of interlocking feedback loops of clock genes that turn each other on and off in a roughly 24-hour cycle. The suprachiasmatic nucleus acts as the conductor, keeping all these cellular clocks synchronized with each other and with the external day-night cycle. When circadian rhythms are disrupted, as in shift work or jet lag, these cellular clocks become desynchronized, with different organs operating on different schedules, leading to metabolic dysfunction, impaired immune function, and increased disease risk. Growth hormone is one of the many hormones whose release follows a circadian pattern coordinated with sleep. During the first period of deep sleep each night, typically within an hour of falling asleep, the pituitary gland releases a large pulse of growth hormone into the bloodstream. This surge is so reliable that growth hormone levels can be used to assess sleep quality. Disrupted or insufficient deep sleep leads to reduced growth hormone release. Growth hormone is crucial for physical restoration. In children, it drives growth in height by stimulating bone elongation. Throughout life, it promotes protein synthesis, particularly in muscle tissue, helping to build and repair muscle fibers. It stimulates the breakdown of fat for energy while preserving the lean body mass. It enhances calcium retention and bone mineralization. It supports immune function by promoting the development and activity of immune cells. The timing of growth hormone release during sleep makes evolutionary sense. During the day, you are active, using energy, breaking down tissues. At night, when you are not eating and need to conserve resources, growth hormone shifts metabolism toward repair and rebuilding. It mobilizes fat stores for energy while directing amino acids toward protein synthesis. The net effect is tissue repair and maintenance without requiring continuous food intake. As you age, both deep sleep and growth hormone release decline in tandem. Older adults spend less time in slow-wave sleep and release less growth hormone. This dual decline contributes to many features of aging: decreased muscle mass, increased body fat, reduced bone density, thinner skin. Whether the decline in growth hormone is a cause or consequence of reduced deep sleep remains debated, but the two are clearly interconnected. Recent research has begun to unravel the neural circuits linking sleep and growth hormone. Scientists have identified specific neurons in the hypothalamus that promote growth hormone release during sleep and discovered a feedback mechanism where growth hormone itself influences wakefulness. When growth hormone levels are high, they activate neurons in the locus coeruleus, a brainstem region involved in arousal and attention. This creates a self-limiting system. Sleep promotes growth hormone release. Growth hormone promotes wakefulness, preventing excessive sleep. This feedback loop may explain why both too little and too much growth hormone can be problematic. Too little, whether from inadequate sleep or pituitary dysfunction, impairs tissue repair and maintenance. Too much can push the brain toward wakefulness, disrupting sleep. The system is balanced to provide the right amount of restoration without interfering with the next day's waking activities. Cortisol, the stress hormone, follows an inverse pattern to melatonin. Cortisol levels are lowest in the evening and during early sleep, rising in the early morning hours and peaking shortly after awakening. This cortisol awakening response helps mobilize energy stores and sharpen alertness for the coming day. Throughout the day, cortisol levels gradually decline, reaching their nadir in the evening. This pattern makes sense from an energy standpoint. Cortisol is catabolic, promoting the breakdown of proteins and fats to release energy. During sleep, when you need to conserve energy and repair tissues, low cortisol creates a permissive environment for anabolic processes like protein synthesis. As morning approaches and you prepare to wake, rising cortisol shifts metabolism toward energy mobilization, readying you for activity. Disruption of this cortisol rhythm has metabolic consequences. Chronic stress can flatten the cortisol curve, maintaining elevated levels throughout the day and night. Sleep deprivation can similarly dysregulate cortisol, with levels remaining high when they should be low. The result is a shift toward catabolism, the breakdown of tissues, at the expense of anabolism, the building and repair of tissues. Over time, this contributes to muscle loss, immune suppression, and metabolic dysfunction. The emotional brain during sleep. Beyond physical restoration, sleep serves crucial functions for emotional processing and regulation. The connection between sleep and emotional well-being has been recognized for millennia, embedded in folk wisdom about not making important decisions when tired or letting angry feelings cool overnight. Modern neuroscience is revealing the mechanisms behind these observations, showing that sleep, particularly REM sleep, plays an essential role in processing emotions and maintaining emotional stability. During waking, as you experience events throughout the day, your brain encodes not just the factual details, but also the emotional tone of those experiences. The amygdala, an almond-shaped structure deep in the brain, responds to emotionally salient stimuli, particularly those related to threat or fear. When you encounter something frightening or upsetting, the amygdala becomes activated, triggering the physiological stress response and tagging that memory with emotional significance. Normally, this emotional tagging is useful. It ensures that you remember dangerous situations and avoid them in the future. But if emotions remain attached to memories at full strength indefinitely, you would be overwhelmed, constantly reliving the emotional intensity of past events. You need some way to preserve the memory, the knowledge of what happened, while reducing its emotional punch. This is one of the functions of REM sleep. During REM sleep, the brain replays memories, reactivating the neural patterns associated with recent experiences. But it does so in a unique neurochemical environment. Unlike waking, when the neurotransmitter norepinephrine is present and active, during REM sleep, norepinephrine levels drop to nearly zero. Norepinephrine is associated with stress and arousal, with the fight or flight response. Its absence during REM sleep creates a safe space where memories can be reprocessed without triggering the full emotional and physiological stress response. This emotional memory reprocessing was elegantly demonstrated in studies where participants viewed emotionally disturbing images before either sleeping or remaining awake. When they were shown the images again after an interval, those who slept, particularly those who obtained adequate REM sleep, showed reduced emotional reactivity. The amygdala's response to the images was diminished. The participants could remember what they had seen, but reported feeling less distressed by it. Those who remained awake or who were deprived of REM sleep maintained their initial emotional response. Further supporting this model, brain imaging during REM sleep shows that while the amygdala and hippocampus, regions involved in emotional memory, are highly active, the prefrontal cortex, which regulates emotions and maintains perspective, becomes less active. This pattern suggests that REM sleep allows emotional memories to be replayed and processed without the cognitive control that might inhibit full emotional expression, but also without the physiological stress response that would reinforce the emotional intensity. The result is what sleep researchers call emotional resolution, the integration of emotional experiences into long-term memory in a way that preserves the knowledge of what happened while reducing the raw emotional charge. You remember the difficult conversation with your supervisor, but wake up feeling less upset about it. You recall the embarrassing social moment, but without the same visceral cringe. Over time and repeated nights of sleep, emotional memories are gradually stripped of their most intense emotional components, allowing you to remember your life without being enslaved by its emotional content. This emotional processing during sleep appears to be essential for emotional regulation more broadly. People who are sleep deprived show increased amygdala reactivity to negative images, a decreased connection between the amygdala and prefrontal cortex, an impaired ability to regulate emotional responses. They report feeling more irritable, anxious, and emotionally reactive. After just one night of good sleep, these changes largely reverse. Chronic sleep deprivation has more serious emotional consequences. It is a risk factor for developing depression and anxiety disorders. People with insomnia have dramatically increased rates of developing major depression. The relationship appears bidirectional. Poor sleep contributes to mood disorders, and mood disorders disrupt sleep, creating another vicious cycle. Understanding sleep's role in emotional processing has implications for treating trauma. Post-traumatic stress disorder is characterized by intrusive memories of traumatic events that retain their full emotional intensity, causing distress and dysfunction months or years after the trauma. Research suggests that PTSD may involve a failure of normal emotional memory processing during sleep. People with PTSD often have sleep disturbances, particularly disrupted REM sleep, preventing the usual overnight resolution of emotional memories. This has led to therapeutic interventions aimed at improving sleep in trauma survivors. Image rehearsal therapy, where people practice reimagining their nightmares with different, less distressing endings while awake, can reduce nightmare frequency and improve sleep quality. Other approaches target the neurochemistry of sleep. Prazosin, a medication that blocks norepinephrine receptors, has shown promise in reducing nightmares in PTSD by allowing more normal emotional processing during REM sleep. Neuroplasticity, the brain that rewires itself. If cellular repair maintains the structure of neurons, and sleep provides the context for emotional and memory processing. Neuroplasticity is the process by which experience actually reshapes the brain's circuitry. For most of history, the adult brain was considered essentially fixed. The circuits laid down during development were thought to be permanent, capable of degradation with age or injury, but not capable of meaningful change or growth. This view has been spectacularly overturned. The adult brain is not fixed but plastic, constantly rewiring itself in response to experience. Every time you learn something new, practice a skill, or even just pay attention to something different, you are reshaping the physical structure of your brain at the level of synapses, the connections between neurons. The fundamental principle of neuroplasticity was captured by Donald Hebb in his famous statement, "Neurons that fire together, wire together." When two neurons are repeatedly active at the same time, the synapse connecting them becomes stronger. This strengthening, called long-term potentiation, involves physical changes at the synapse. More neurotransmitter receptors are inserted into the receiving neuron's membrane, making it more sensitive to signals from the sending neuron. The synapse itself may grow larger with more contact area between the neurons. New synapses may even form, creating additional pathways for communication. This synaptic strengthening is how learning happens at the cellular level. When you practice playing a musical instrument, neurons in the motor cortex that control finger movements fire in specific patterns. With repeated practice, the synapses connecting these neurons strengthen, making those patterns easier to activate. The initially difficult passage that required intense concentration gradually becomes automatic, executable without conscious thought, because the neural circuit has been physically reinforced. Conversely, synapses that are rarely used weaken and may eventually be eliminated, a process called synaptic pruning. This might sound like a loss, but it is actually essential for efficient brain function. A brain that never pruned unused connections would become cluttered with irrelevant pathways, unable to distinguish important patterns from noise. By strengthening frequently used synapses and weakening rarely used ones, the brain sculpts its circuits to reflect what is actually relevant in your environment and behavior. This pruning is particularly important during development. A newborn's brain contains more synapses than an adult's, with neurons making promiscuous connections to many potential partners. Through experience and learning, some connections are reinforced while others are eliminated. The result is a more organized, efficient brain with stronger, more reliable circuits for important functions. But plasticity continues throughout life. Brain imaging studies have shown structural changes in adults learning new skills. London taxi drivers, who must memorize the complex layout of the city's streets, show enlargement of the posterior hippocampus, a region involved in spatial memory. The longer they have been driving taxis, the larger this region becomes. Musicians show structural changes in motor and auditory regions corresponding to their instrument. Jugglers develop enlarged regions involved in visual motion processing. These are not just abstract changes, but have functional consequences. When experienced meditators practice focused attention, they show enhanced activity in prefrontal regions involved in attention control and reduced activity in regions involved in mind wandering. This pattern becomes more pronounced with years of practice, suggesting that meditation literally reshapes attention networks in the brain. Neuroplasticity also underlies recovery from brain injury. When a stroke damages brain tissue, the functions controlled by that tissue are initially lost. But over weeks and months, some recovery often occurs as remaining brain regions take over the lost functions. This reorganization involves the strengthening of existing but weak connections and the formation of new synapses, allowing undamaged areas to assume roles they previously did not perform. The extent of recovery depends on many factors. Age matters. Younger brains are generally more plastic and recover more completely. The size and location of the damage matter. Smaller, more peripheral damage is easier to compensate for than large lesions in critical regions. But perhaps most important is rehabilitation. Intensive, focused practice of lost functions drives the neuroplastic reorganization that underlies recovery. Simply waiting passively does not produce the same gains. This has transformed stroke rehabilitation from a fatalistic acceptance of permanent disability to an active pursuit of recovery through targeted therapy. Constraint-induced movement therapy, where the unaffected limb is restrained to force use of the affected limb, can drive remarkable recovery by forcing the brain to engage surviving circuits. Mirror therapy, where patients watch their unaffected hand in a mirror while attempting to move the affected hand, can reduce pain and improve function, perhaps by recruiting visual motor circuits to support movement. Neuroplasticity is not limited to motor functions. Language recovery after stroke involves similar principles. Intensive speech therapy drives reorganization of language networks, with remaining brain regions taking over functions of damaged areas. Even in severe aphasia, where stroke damages primary language areas, some recovery is often possible through reorganization of secondary language areas in the same or opposite hemisphere. Beyond recovery from injury, neuroplasticity can be harnessed for cognitive enhancement. Cognitive training programs, where people practice specific mental tasks like working memory exercises or attention tasks, can produce improvements not just in the trained tasks, but in related cognitive abilities. The gains are accompanied by measurable changes in brain structure and function, with enhanced activity in relevant brain regions and strengthened connectivity between them. However, the limits of such training are debated. Some studies show impressive gains that transfer to everyday cognitive tasks. Others find benefits limited to the specific trained tasks with little generalization. The field is working to understand what types of training produce the most meaningful benefits and for whom. Age, baseline, cognitive ability, motivation, and training intensity will appear to matter. Sleep and neuroplasticity, a crucial interaction. The relationship between sleep and neuroplasticity is bidirectional and essential. Sleep supports neuroplasticity, and neuroplasticity shapes sleep. Learning during the day drives changes in sleep that consolidate that learning. Sleep deprivation impairs the neuroplastic changes that underlie learning and memory. When you learn a new skill or encode new information during the day, your brain is not just passively recording that information. It is actively changing its structure, strengthening some synapses, weakening others, growing new dendritic spines where new synapses will form. But these initial changes are fragile. Without consolidation, they will decay. Sleep provides the crucial consolidation window. During sleep, particularly during slow-wave sleep, the brain replays patterns of neural activity from the day. Sequences of neural firing that occurred during learning are reactivated in fast-forward, sometimes called replay. This replay is not random, but specifically involves the circuits engaged during learning. If you spend the day learning to navigate a maze, hippocampal neurons that encode spatial positions in that maze will reactivate during subsequent sleep, replaying your path through the maze. This replay serves multiple functions. It strengthens the synapses involved in the learned task, cementing the neural changes initiated during learning. It integrates new information with existing knowledge, connecting new memories to related old memories. It extracts general principles from specific experiences, a process that appears particularly important during REM sleep. The connection between learning and subsequent sleep is so strong that the amount and quality of slow-wave sleep after learning predicts how well that learning will be retained. People who get adequate slow-wave sleep after learning show better memory performance than those who are sleep deprived, even when tested days later. The difference is not just in recall, but in the quality of memory. Sleep consolidated memories are more resistant to interference, more flexible, and more integrable with other knowledge. Sleep deprivation before learning also impairs memory formation, but through different mechanisms. Without adequate sleep, the brain's capacity to encode new information is reduced. The hippocampus, crucial for forming new memories, shows reduced activity during attempted learning when people are sleep deprived. The consolidation that should happen during the subsequent sleep is also impaired because the initial encoding was weak. This bidirectional relationship means that optimizing either learning or sleep can enhance the other. Engaging in challenging mental activities during the day, learning new skills, having novel experiences, these enhance subsequent slow-wave sleep, which in turn better consolidates that learning. Prioritizing sleep improves the brain's capacity for learning the next day, creating a virtuous cycle. The molecular mechanisms linking sleep and synaptic plasticity are being elucidated. During waking, synapses throughout the brain generally strengthen as you learn and experience the world. If this strengthening continued unchecked, synapses would become saturated, unable to strengthen further, and neurons would become hyperactive, requiring excessive energy. Sleep provides a period of presynaptic downscaling, where synapses are globally weakened, restoring the capacity for further learning. But this downscaling is not uniform. Synapses that were strongly activated during learning, that are important and relevant, are protected from downscaling or even strengthened. Weak, irrelevant synapses are preferentially weakened or eliminated. The result is a sharpening of neural circuits. Important signals are amplified, noise is reduced, and the brain emerges from sleep with a refreshed capacity for learning. This synaptic homeostasis hypothesis explains many features of sleep. It accounts for why sleep is necessary after learning. It explains why sleep deprivation impairs cognitive function. Without synaptic downscaling, neurons become hyperexcitable and circuits lose their precision. It even explains some features of sleep architecture. The slow waves of deep sleep may be the signature of synchronized synaptic downscaling across large populations of neurons. Aging, renewal, and the limits of restoration. As we age, all of these renewal systems, cellular repair, sleep architecture, autophagy, neuroplasticity, gradually decline. DNA repair becomes less efficient, allowing mutations to accumulate. Autophagy weakens, permitting damaged organelles and protein aggregates to build up. Sleep becomes more fragmented and contains less deep, slow-wave sleep, reducing both physical restoration and memory consolidation. Neuroplasticity persists, but at reduced levels, making learning more effortful and recovery from brain injury less complete. Yet, this decline is not uniform or inevitable. Significant individual variation exists in how rapidly these systems deteriorate. Some people maintain robust repair and renewal mechanisms well into old age, while others show premature decline. Understanding what drives these differences could provide targets for interventions to slow aging and extend health span. Genetics plays a role. Some people inherit more efficient DNA repair systems or more robust autophagy machinery, but lifestyle factors may be equally or more important. Regular exercise enhances autophagy, improves sleep quality, promotes neuroplasticity, and reduces oxidative stress. A nutrient-dense diet provides the building blocks needed for repair and the antioxidants that reduce damage. Cognitive engagement and social interaction promote neuroplasticity and cognitive reserve, the brain's resilience against age-related decline. Caloric restriction, the most robust intervention for extending lifespan in laboratory animals, appears to work in part by enhancing cellular repair mechanisms. When nutrients are limited, cells activate stress response pathways that upregulate DNA repair, boost autophagy, and enhance antioxidant defenses. The same pathways can be activated by intermittent fasting or by drugs that mimic some effects of caloric restriction. Emerging therapies aim to directly enhance cellular renewal. Senolytic drugs selectively kill senescent cells, those dysfunctional cells that accumulate with age and secrete inflammatory factors that damage their neighbors. Removing these cells in animal models improves multiple aspects of health and extends lifespan. Early human trials of senolytics show promising results, with improvements in physical function and reduced markers of inflammation. Other approaches target autophagy directly. Compounds that activate autophagy, from rapamycin to spermidine to urolithin A, are being tested for their potential to slow aging and prevent age-related diseases. The goal is not to stop aging completely, an impossibility given the fundamental role of entropy in biological systems, but to compress morbidity, keeping people healthy and functional for a larger fraction of their lifespan. Stem cell therapies represent another frontier. By replacing cells that have been lost or damaged, stem cells could theoretically restore tissue function. But significant challenges remain. Ensuring that transplanted cells integrate properly, survive long-term, and don't form tumors is difficult. Understanding how to direct stem cells to differentiate into specific cell types and how to support them once transplanted requires much more research. Perhaps the most promising direction is combination approaches that address multiple aspects of aging simultaneously. Exercise, good nutrition, adequate sleep, cognitive engagement, and social connection together create an environment that supports cellular and neural health across all systems. Adding targeted interventions like autophagy, enhancers, or senolytics on top of this foundation may produce synergistic benefits greater than any single intervention alone. The emotional foundations of renewal. Beyond the molecular and cellular mechanisms, renewal has an emotional dimension. Stress, chronic and unrelenting, suppresses cellular repair, disrupts sleep, impairs neuroplasticity, and accelerates aging. Chronic stress keeps cortisol levels elevated, shifting metabolism toward catabolism and inflammation. It disrupts sleep architecture, reducing slow-wave sleep and fragmenting sleep continuity. It impairs hippocampal neurogenesis and can even cause atrophy of the hippocampus over time. The mechanisms linking stress to impaired renewal are multiple. Elevated cortisol directly inhibits protein synthesis and promotes protein breakdown. Stress hormones increase oxidative stress and inflammation, creating more cellular damage, while simultaneously suppressing repair mechanisms. Stress disrupts the hypothalamic pituitary adrenal axis, the hormonal system that coordinates stress responses, leading to dysregulation of circadian rhythms and sleep-wake cycles. Conversely, positive emotions and psychological well-being appear to support renewal. People who report higher levels of life satisfaction show better sleep quality, more robust immune function, and slower biological aging as measured by biomarkers. Social connection, a sense of purpose, and engagement in meaningful activities are all associated with better health and longer lifespan. The mechanisms here are less well understood than those linking stress to disease, but some pathways are emerging. Positive emotions may reduce inflammation and oxidative stress. Strong social connections may buffer against stress. preventing its damaging effects. A sense of purpose may promote health behaviors like regular sleep, exercise, and good nutrition, which in turn support cellular renewal. The vagal nerve, which connects the brainstem to the heart, gut, and other organs, may play a role. High vagal tone, the strength of vagal nerve activity, is associated with better emotional regulation, lower inflammation, and better health outcomes. Practices like meditation, slow breathing, and yoga increase vagal tone and may promote renewal through this pathway. Emotional regulation itself, the ability to manage and modulate emotional responses, appears important for health. People who can effectively regulate their emotions show reduced physiological stress responses, better sleep, and lower rates of mental and physical illness. The prefrontal cortex, which regulates emotions, also regulates stress responses and interacts with systems controlling sleep and circadian rhythms, providing a neural basis for these connections. This suggests that psychological interventions could support physical renewal. Cognitive behavioral therapy for insomnia improves not just sleep, but also immune function and inflammatory markers. Mindfulness-based stress reduction reduces cortisol, improves sleep, and enhances emotional regulation. These are not just mental benefits, but physical ones, reflecting the deep integration of mind and body in renewal processes. The daily practice of renewal. Understanding the mechanisms of renewal suggests practical approaches to support these processes in daily life. Sleep should be prioritized, not treated as optional. 7 to 9 hours for most adults, with consistency in timing to support circadian rhythms. The bedroom should be cool, dark, and quiet. Electronic devices should be avoided for at least an hour before bed to prevent blue light from suppressing melatonin. Alcohol, despite its sedative effects, disrupts sleep architecture and should be minimized. Physical activity supports renewal through multiple pathways. Exercise enhances autophagy, particularly in muscle tissue. It promotes neuroplasticity and neurogenesis in the hippocampus. It improves sleep quality, particularly deep, slow-wave sleep. It reduces inflammation and oxidative stress. The type of exercise matters less than consistency. 30 minutes of moderate activity most days provides substantial benefits. Nutrition provides the building blocks for cellular repair. Adequate protein ensures availability of amino acids for protein synthesis. Micronutrients, like B vitamins, support DNA repair. Antioxidants from colorful vegetables and fruits reduce oxidative stress. Omega-3 fatty acids reduce inflammation and support neuronal membrane integrity. Fiber feeds beneficial gut bacteria, which produce metabolites that support health throughout the body. Intermittent fasting, whether time-restricted eating, where food intake is limited to an 8 to 12-hour window, or periodic longer fasts, activates autophagy and other cellular stress responses that enhance repair. The optimal approach varies individually, but avoiding late-night eating and allowing a 12 to 14-hour overnight fast appears beneficial for most people. Cognitive engagement, learning new skills, practicing challenging mental tasks, these drive neuroplasticity and build cognitive reserve. The specific activity matters less than that it is novel and challenging. Learning a language, playing a musical instrument, dancing, strategic games, all can promote brain health when practiced regularly. Social connection supports renewal through stress buffering and by providing meaning and purpose. Chronic loneliness is as damaging to health as smoking or obesity. Cultivating relationships, participating in communities, helping others, these behaviors support emotional and physical health. Stress management practices, whether meditation, yoga, time in nature, engaging in hobbies, or simply taking breaks, help maintain the balance needed for optimal renewal. Chronic stress undermines all renewal systems. Finding sustainable ways to manage stress is not a luxury, but a necessity for long-term health. Looking forward, the science of human renewal is revealing a picture of the body as a remarkably self-maintaining system, constantly repairing damage, clearing waste, rebuilding structures, and adapting to challenges. These processes are not separate from consciousness or experience, but deeply integrated with how we think, feel, and interact with the world. Sleep is not lost time, but essential time for cellular housekeeping, emotional processing, and memory consolidation. Emotions are not just mental phenomena, but physiological states that either support or undermine renewal. Learning and new experiences are not just intellectually enriching, but physically reshape the brain, building cognitive reserve that protects against aging. This understanding empowers, but also obligates. You are not a passive victim of aging or disease, but an active participant in your own renewal. The choices you make about sleep, activity, nutrition, stress, and engagement shape how your renewal systems function. Over years and decades, these choices accumulate, determining not just how long you live, but how well you live. The frontier of renewal research is vast. Scientists are working to understand how to enhance autophagy without triggering unwanted side effects. They are investigating how to safely promote neuroplasticity to aid recovery from brain injury and prevent cognitive decline. They are developing interventions to remove senescent cells, enhance sleep quality, and support cellular repair mechanisms. Some of these interventions will work, others will fail or reveal unexpected complications. The path from laboratory discovery to clinical application is long and uncertain. But the fundamental principle is clear. The body possesses extraordinary renewal capacities, and supporting these capacities offers a path toward longer, healthier lives. The renewal that never stops. As you read these words, your cells are reading their own instruction manuals, repairing damaged DNA using templates preserved through billions of years of evolution. Molecular chaperones are rescuing misfolded proteins, giving them another chance at function. Autophagosomes are engulfing damaged mitochondria, clearing them away before they can poison the cell. If you are tired, adenosine is accumulating in your brain, building sleep pressure that will eventually drive you to rest. When you do sleep, growth hormone will flood your body, promoting tissue repair. Your glymphatic system will clear metabolic waste from your brain. Your amygdala and hippocampus will replay emotional memories in a neurochemical environment that strips them of their most intense emotional charge. Tomorrow, when you learn something new, synapses will strengthen, dendrites will grow new spines, and neural circuits will reorganize to incorporate that learning. Experiences will reshape the physical structure of your brain, building the person you are becoming from the person you were. This is the miracle of human renewal. It never stops. Every moment, every breath, your body is engaged in the grand project of maintaining itself, of resisting entropy, of rebuilding what time and circumstance degrade. You are not the same person you were 7 years ago, at least not physically. Nearly every cell in your body has been replaced, rebuilt from the materials of the world, transformed through biological alchemy into you. And yet, you persist. Your memories remain, encoded in patterns of synaptic connections that have been maintained through continuous molecular turnover. Your sense of self continues, a stream of consciousness flowing through a body in constant flux. You are a pattern preserving itself through perpetual renewal, a process that has continued unbroken in your lineage for billions of years, back to the first replicating molecules in ancient oceans. The science of renewal tells us that we are not solid objects, but processes, not fixed, but dynamic, not passive, but active participants in our own becoming. Every night, when you sleep, every time you learn, every moment of emotional experience, you are engaging in the ancient work of renewal that makes life possible. Understanding this changes how we relate to ourselves. We are not machines that inevitably wear out and break down, but biological systems with remarkable capacities for repair and adaptation. We are not victims of aging, but partners with our renewal systems, able to support or undermine them through our choices. We are not separate minds riding around in meat vehicles, but unified mind-body systems where psychological states influence physical health and physical health shapes psychological experience. This is the deeper meaning of renewal, not just the molecular mechanisms of repair, but the integration of all these processes into a coherent whole. The remarkable achievement of maintaining a complex, organized system in a universe that tends toward disorder. You are that achievement, that ongoing triumph of order over entropy, of renewal over decay. As you move through your days and nights, as you sleep and wake, learn and remember, feel and heal, you are participating in the most fundamental process of life, the perpetual renewal that makes existence possible. It is happening now, in this moment, as it has happened in every moment of your life and every moment in the lives of your ancestors, stretching back to the origin of life itself. This is your inheritance and your gift, the capacity for renewal, written into every cell, every synapse, every heartbeat. Use it well. Support it with sleep and movement, with learning and connection, with purpose and presence. And know that every moment of care you give to these renewal systems is an investment in all the moments to come, a recognition of the precious gift of being a self-renewing being in a universe of constant change. If you found this journey into human renewal valuable, consider subscribing to Dream Lab Knowledge and sharing this with others who might benefit from the understanding how their bodies and minds maintain themselves across the lifespan. Your support allows us to continue exploring the fascinating science of what makes us human. Sweet dreams, and may your renewal systems work their quiet magic as you rest tonight.